Hello guys, welcome to my channel. So as you know, we are studying about the protein structure. We studied about the protein primary and secondary structure. Now we'll see protein tertiary and quaternary structure. Quaternary structure. Right. So what is protein tertiary structure? So the overall three-dimensional arrangement. So that is overall three-dimensional arrangement of all atoms in a protein. It is going right. So overall arrangement of all atoms in a protein is referred to as the protein's tertiary structure. Tertiary protein tertiary structure. So this is the tertiary structure. So the secondary structure that we saw, that refers to just the spatial arrangement of amino acid residues that are adjacent in a in a segment of polypeptide, right? So if there is a polypeptide segment, so the arrangement of amino acid residues, how it will be arranged, that was secondary structure. Like it has like hydrophobic, it folded because of weak interactions between hydrogen bond or the van der Waals interaction, it folds, right? But the tertiary structure includes longer aspects of amino acid sequence, not that much shorter, but it includes a very longer aspects of amino acid sequence, and this it's in the three-dimensional arrangement of all the atoms in a protein. That is tertiary structure, right? And so the quaternary structure, like some protein contain two or more separate polypeptide chain. So there are some kind of protein that have not just a single polypeptide chain, but it has two or more separate polypeptide chain or subunits, which may be identical or different, right? So the arrangement of this protein subunits. So the arrangement of this protein subunits is in three-dimensional complexes constitute quaternary structure. So what is quaternary structure? So some protein contain, I will write it just down here. Protein contains two or more polypeptide chains or subunits, which might be identical or different, but the arrangement of this protein subunits are in three-dimensional complexes is constitutes the quaternary structure, right? So to make it more simpler, we can divide this higher level structure into two groups. So that is, first group we can say fibrous protein, right? So the fibrous protein, in their polypeptide chains arranged in long strands or sheets. So write it down here. Polypeptide chain arranged in long strands or sheets, right? So this was the one type, and the second type is globular protein. Right. So in globular protein, the polypeptide chain folded into a spherical or the globular shape. So the polypeptide chain is folded or globular in shape. Alright. So these are the two groups. So that are structurally distinct. So the fibrous protein consists largely of a simple single type of secondary structure, right? As it has a long strands, and that tertiary structure is relatively simple. But for the globular protein, often contain a several types of secondary structures, and the tertiary structure is more complex. So that these two groups also differ in functionality as well, right? The structure that provides support, shape, and external protections to vertebrates are made up of fibrous protein, whereas the enzymes and regulatory proteins are made up of globular protein, right? So they both, their functions are also different, right? Because this gives support, shape, and external protection, and these are the most enzymes and regulatory proteins, right? So first we'll see the fibrous protein. So the fibrous protein share properties that give strength, right? The strength and flexibility to the structure in which they occur, right? In each case, the fundamental structure unit is a simple repeating element of secondary structures. So all fibrous protein are insoluble in water, right? So I will just write one and just write the main points. So all fibrous protein are insoluble in water, and a property conferred by a high concentration of hydrophobic amino acid residues. So this fibrous protein will have high concentration of hydrophobic amino acid residues both in the interior of the protein and on its surface as well. That's why the fibrous protein are insoluble in water. So these hydrophobic surfaces are largely buried, as many similar polypeptide chains are packed together to form supramolecular complexes. So these hydrophobic surfaces of the fibrous protein are largely buried inside, as many polypeptide chains are packed together to form an elaborate supramolecular complexes. Right, we'll see few of the examples for the fibrous protein that will illustrate the relationship between the protein structure and the biological function for this fibrous protein. So the first example is the alpha keratin. All right, the first one is alpha keratin. So the alpha keratin characteristics is tough, insoluble protective structure of variable hardness and flexibility. So the characteristic, I'll write it down here. So the alpha keratin is tough, insoluble protective structure. Protective structure of varying hardness and flexibility. Okay. So I will write it down here. Hardness and flexibility. All right. So the alpha keratin has evolved for strength. So this alpha keratin are found in mammals. This protein constitutes almost the entire dry weight of a hair wool. So the example that where the alpha keratin are found, so they are found in nails, quills of the porcupine. Right, so these are the examples that the alpha keratin is found, and this protein constitutes the entire dry weight of the hair or the wool or the nails, claws, quill, and also it's found in much of the outer layer of the skin, and much of the outer layer of the skin, layer of skin. All right. So the alpha keratin are part of a broader family of proteins that are called intermediate filament protein. So these are the part of a broader family protein. So, right, so these are the part of intermediate family and protein. That's the family of, that's where the alpha keratin belongs. So other intermediate filament proteins are like that are found in cytoskeleton of animal cells, and all the intermediate filament protein have a structural function and share the structural features exemplified by the alpha keratins. So the alpha keratin helix is a right-handed helix, alpha helix, the same helix that are found in many other proteins, right? If you remember that we studied about the left and the right alpha helixes. So the alpha keratin belongs to the right-handed alpha helix, right? And that was discovered by Francis Crick and Linus Pauling, correct? So the two strands of alpha keratin oriented in a parallel are wrapped about each other to form a super twisted coiled coil. Right, so how this alpha keratin is used for this hair, nails, claws, or quills. So we did the left and the right handed alpha helix. So this is regarding the right hand alpha helix. So I will just draw a two-dimensional picture here. For example, this is just a one alpha helix, all right, and there will be another one overlapping. So just like a rope, if you see a rope, right? So it has something like this. And there will be right, so like intermingled like a rope, right? So the two strands of alpha keratin oriented in a parallel, so with the amino terminal at the same end, are wrapped about each other to form a super twisted coil, right? So the this super twisting amplifies the strength of the overall structure, like as the rope, how it's super twist and it's minded in the coil to describe the strength of the rope. So just at the same as a strand are twisted to make a strong rope of alpha keratin. And the helical path of the super twist. So the super twist, the helical path of the super twist is left-handed, opposite in a sense to the alpha helix, right? And the surface is, so the surface where two alpha helices touch. So for example, I will draw a bigger picture here. Maybe like this, something like this. But the surface is where this two alpha helices touch are made up of hydrophobic amino acid. So in between, this are made up of hydrophobic amino acid where they touch each other, amino acid residues, and their R group are meshed together in a regular interlocking pattern. Right. So for example, this is, I will just write it down, made up of hydrophobic amino acid, and their R group are meshed together in an interlocking pattern, right? So this permits a close packing of a polypeptide chain within the left-handed super twist. So this hydrophobic amino acid residues comes from, so that's why the alpha keratin is rich in the hydrophobic residues that are ALA, alanine, valine, right? LEU, leucine, ILE, isoleucine, right? MET, methionine, and PHE, this phenylalanine, right? So these are the amino acid residues that are hydrophobic, and that's why they are found in this alpha keratin. So this intertwining structure of the two alpha helical polypeptide is an example of a quaternary structure. So this is a quaternary structure, and the coiled coils this type are common structural elements in the filamentous protein and in the muscle protein. For example, that is myosin as well. Myosin, this is also found in the protein Myosin and the quaternary structure of this alpha keratin can be a quite complex, because the many coiled coils can be assembled into a large supramolecular complexes, such as an arrangement of alpha keratin that forms the intermediate filament of hair. So how the intermediate filament of hair is formed? So I'll just show you a diagram. So for example, so this is one alpha keratin, right? So there will be like the second one here. I will just draw a random picture, like just imagine that that's a rope and that's the second one. And so this two, for example, or three, that will form a filament, right? So this two will form a protofilament. When there will be like more than more than 4, 5, 6, 7, 8, so many of this alpha keratin filament, so there it will be termed as protofibril, right? So this will be termed as protofibril, right? So this form the protofibril about more than like 4, 5, 6, 7. So about 4 protofibrils. So that is so about 4 protofibrils. So that is around like 32 strands of alpha keratin. So in all combine, this all will combine together to form an, so that will form an intermediate filament, right? So about 4 protofibrils. So in 4 protofibrils there will be about 32 strands of alpha keratin in all, and that will combine to form an intermediate filament. So you can see a picture on your screen. So that shows a structure or cross section of a hair, and you can see in that cross section, so there is a cell, and then that's an intermediate filament. Then there is protofibril that we talked about. Then there is the protofilament, and from the protofilament there is this two coiled alpha helix structure of the alpha keratin. So this is how it is done. Right, so the strength of this fibrous protein is enhanced by the covalent cross links between the polypeptide chain in the multi-helical ropes and between the adjacent chains in the supramolecular assembly, right? So the strength of this will be depending on the covalent cross links between the polypeptide chains. And in alpha keratin, the cross links stabilizing the quaternary structure, right, are the disulfide bonds. So in this alpha keratin, the cross links that stabilize the structure are the quaternary, are the disulfide bond. Here, quaternary structure, the stabilizing is disulfide bond. And in the hardest and the toughest alpha keratin, such as the rhinoceros horn, like the, for example, maybe you can see here, for example, in rhinoceros horn, right? So the toughest and the hardest alpha keratin are found, and in that there are the amino acid residues of cysteine involved in the disulfide bond. So there are like up to almost 18% of the residues that are cysteine in the rhinoceros horn. All right, that involves the disulfide bonds. Right. So this was one example of the fibrous protein, that is alpha keratin. So I will just remove this and we'll talk more about this.